video project, I wanted to use sources that were either affiliated with the government or something to do with any university to maximize the credibility and make sure that my sources are the best. And then I also used the notes that we were working on in class. And I'm gonna start off with the structure of DNA. So I'm gonna draw a simplified model because my drawing isn't really the best. And so we have here in the middle the nitrogen bases. In the structure of DNA, which is a double helix model, although this is a more simplified version, there are the two sides of the DNA, or the DNA backbone, which usually spirals around the center. It's made up of deoxyribose connecting to phosphate, and then they are all connected together. In the middle here, you see the nitrogen bases, which are adenine, guanosine, which are purines, meaning they are double ring structures. And then there's thymine and cytosine, which are pyrimidines. These are represented by the first letter in the word. And so guanosine always pairs with cytosine, and then adenine always pairs with thymine, because that's just the way it works. They attract together, and then what differs is the way they are connected, and that's where the mutations come in. And in the middle here, connecting the two sides of the DNA structure are the hydrogen bonds. Now to move on to how DNA makes protein, we're first gonna I'm first gonna talk about transcription and translation. So here there's the original DNA strand, and then this is the messenger RNA, and then here we have the amino acids. So with the original DNA strand, we have this sequence of the nitrogen bases, and then we transcript it into the mRNA, which give us different codons, which are groups of 3 nitrogen bases, which we can then translate into amino acids, and these groups then form proteins. The goal of this translation is so that the cell can tell the organelles that make the protein exactly how to link up the amino acids in order to make the right protein. And with the variations in the mRNA, which can cause mutations, there are deletion, insertion, and substitution mutations. Deletion is, say for example, we take out this U, making the new codon AGG, GCU, CUU, AG blank, because although there's a continuation, we don't have it. This changes the amino acids produced, and it changes the frame, making it a frame shift mutation. And then if we were to bring back the U here, and say we were to add a C here, this would also change the frame and make a frame shift, because it changes the frame of the mRNA, but also changes the amino acids. With the substitution mutation, say we substituted the C here with a U, we then changed this codon, and it could either be silent, meaning that this would result in the same amino acid, or it could be missense, meaning that it would change the amino acid, or it could result in a stop codon, making it a nonsense mutation. These mutations are not necessarily bad, but say a nonsense mutation happens very early on, it could cause devastating effects because it has changed a lot from the original DNA strand. Something, however, changing a silent mutation would not change anything, and maybe one codon being changed could be beneficial, not harmful.